Greetings. This tutorial is meant to help you get the most out of Xenbase for your molecular biology research using the Xenopus model organisms. As of December 2013, Xenbase has issued a major update to the website, and this tutorial will help you find the new content that is available on the site now, as well as help you find the items that have moved. And because this tutorial replaces an existing tutorial about how to navigate the site, I will also provide general tips for that, as well as highlight features that are very powerful but are underused on the site. If you are a returning user, the most prominent change that you'll notice is, besides the general layout, this image carousel here at the top center of the page. These images are linked to news items that will feature information about notable papers, awards that have been issued to Xenopus researchers, funding opportunities or program announcements, new Xenbase features, and scientific meeting announcements, as well as other items of importance to the Xenopus community. Also new to the site are instructional tooltips. So if there is a link and a title listed, but it's not terribly clear, if you mouse over it for a moment, you'll get this information bubble that will help you understand what you might be going to before you commit to the click. There's also the option to follow us on Facebook and Twitter. Those have been useful already to Xenopus researchers at meetings, for example, as well as for finding updates from Xenbase. Also, there are 3 new important sets of data available to you. The first is our highly anticipated antibody feature. This is an actual database integrated with the Xenbase database. It can be found by either navigating to the main content area here and clicking on antibodies, or else using the reagents and protocols title and clicking the first entry antibodies. Here at the interface for the antibodies, you can search by various methods, but it defaults to search all. Here you can search for anything that you want, or you can hit browse all as an example. You can make it do that, and you can see the list of antibodies listed by gene symbol and unique identifier AB, 1, 2, 3, and so forth, as well as common names, synonyms, whether or not it's monoclonal or polyclonal, and the reported usages, what applications it's used for. Once you click on an antibody, you'll come to a main page for it. Here you'll find, again, the name, the common name, synonyms, as well as the different things that it will react to. This can either be gene symbols, Go terms, or Xenopus anatomy ontology terms to help you find tissue-specific antibodies. The other data that are captured are down here, and of particular note, most of the time to find information about how to get the antibody, that's under description, and we'll have a note that says supplied by either the manufacturer or who made the homemade antibody. Also, most of the antibodies will have Xenbase attributions here under this tab. If I go back to the main page, I'll point out the other 2 features that are not yet integrated with the database but are available to you in list form until we can get them linked up. Those are the Morpholino Wiki. So again, under reagents and protocols, you can find a list of morpholinos, and this will take you to a wiki page, and you'll have to click on this, which is a Google document that we've co-opted, and here you can find information about the symbol of morpholinos, in this column, the sequence, the article number in Xenbase, so XB-art- then the number, and other information. Finally, there is, under expression, there's a new catalog of microRNAs provided by the data from Grant Wheeler and Mike Gilchrist. And here you have the sequence of the microRNAs, as well as images and links to Xenbase. So currently we only have in here the microRNAs for which we have expression data. So all of them have a camera icon. But for example, if you clicked on this one, you'll find both the images of the expression determined by in situ, as well as literature. And if we go back, a link to Xenbase will provide other information through their database. That's the most prominent new features. Other things have moved from the website's previous layout. Most notably, the search bar is now up here at the top. It behaves in precisely the same way. There are many different search genres. It defaults to genes, though. There are still two ways to find most of your content. The first is in the title bar. That follows you throughout the site and has subheadings to help you find the more specific information that you're interested in more directly. There's also the main content area here, which essentially displays those title bar items in list form in a general area. And finally, the information from the sidebar that was previously on the left has moved to the right, and we have added into it another display of the announcements, including an archive found here. There's information for general users on why to use Xenbase as a model organism, other features, and the existing links are actually accordioned down here at the other resources heading. You can find those links there, mostly to external sources. So I'd like to now transition to essentially the content of our previous how-to videos, which is how to navigate. Importantly, we should talk about how to use the searches. So all of our searches rely on a controlled vocabulary, so there has to be an existing database entry for the search to return anything. So for example, genes, if you search for genes, only terms that represent an actual gene symbol, gene title, or a synonym will produce a result. So if I search for expra, which is a synonym of brachyury, and I find these auto-populated lists here, I can either click on the item here if I know that I want exactly expra, or I can hit search, which will send me to a disambiguation page. From these, I would have to select, and I would then go to the gene page. Sorry, a little display error there. So the gene page is where most of our content is, and it is organized into these larger sets that are organized by tabs. So you have to browse for your most information here, but there are direct links to the gbrowse, sequences, information about the molecule's importance to Z's, and so forth. But if you wanted to find other content, you might go to the different tabs. Say here at the expression tab, you can see the different images generated by various screens and or annotations from the literature, of which for which Expra has a lot of content. So each of these is a literature image. Okay. The general trick of clicking on the Xenbase title, taking you back to the front page, still works with a new page, with a new website. As an illustration, let me just show you that if you did the same search here and clicked on the controlled vocabulary item, you would go directly to that gene page. One thing of note is that we use the official gene symbols that are handed down from the Human Naming Committee, and the official gene symbol is listed here. So for Expra, it's actually T, and only T, and we encourage you to use the actual official symbol in your publications and communications. Okay. Still, concerning the searches, because this is still the main way to navigate to the content that you need, Note that the controlled vocabulary that is available for a search is specific to what search you have set up. So if you wanted to look for a particular anatomy item and you searched using the anatomy items heading, you could find an auto suggestion for whichever structure you are interested in. So if I type in pronephric kidney, I can select it here, and it will take me to that page. Okay, and here you can find information about what the pronephric kidney is, including on the expression tab, images that have been tagged by curators that show a gene is expressed in that structure. If I go back and I still have anatomy items selected for my search, it will not respond to other irrelevant controlled vocabulary terms like expra being a gene name or a gene symbol, synonym rather. It is not relevant to the anatomy items database and will not find it. So be careful what you've selected and what you put into that search. Finally, there's one other search that I will point out for you in particular because it is particularly useful for searching for Pubmed Ids, which is not directly available on the search bar otherwise. So if you select Xenbase with Google, you can then find information by Pubmed ID. So enter Pubmed ID here, and it will find the relevant article for you. As an example, I have that one prepared. It should take us to a nice paper by the Zorn Lab. Okay. Finally, I'd like to talk about the underused features of Xenbase, and I will present those only in list form. There are still others highlighted in separate tutorials. So for the Xenopus community, we appreciate it if you would send us your large-scale data. You can do so, and it will be displayed here at the Xenbase FTP. You can find out different features about your gene and the surrounding DNA or chromosomal structure here in the genome browsers, which we are in the process of updating. The most recent Xenopus laevis is actually available to us now. You can also upload your own tracks to this to do your own analysis. Next, we have manually curated a list of protocols that are commonly used by the community. They are here under Reagents and Protocols and Protocols Wiki. Here you'll find that list. These are manually curated and are both up to date and in principle the best practices of the community. So this is a good place for those. There's also a host of static content to learn about the Xenopus system, including atlases and just information about development in general. In particular, the new and Coop and Faber stages are available here under the Anatomy and Development heading. And here are these images. Next item, there is a very powerful literature search called Texpresso, which has all of the text from each section of every article in Xenbase preloaded into a queryable database, and whatever terms you put in, it will find, whether it's your gene or your sequence or your random text, it will find other items associated with that in the same sentence or in the same paragraph. And so that's an incredibly powerful tool. There is a separate tutorial on how to use that. There are two more items. The first is jobs. So we encourage not only those people looking for a job, but those looking for staff to use the job postings under community. You can send them your description to us if you can't upload that yourself. And finally, we would like to urge you to use the Contact Us button and send us emails whenever you find a bug or have a suggestion for content or anything else you want Xenbase to do for you. or if annotations are not proper, we can correct them, or we can prioritize your favorite new paper for curation. That should be it for this tutorial. Please look for updates to the other tutorials on advanced searches and the main content of the page. We hope you enjoy the new site, and again, please contact us with any feedback.